So here we have our 4 plates of E. coli growth. The first one is LB amp minus P-glow. This shows that bacteria is not resistant to ampicillin naturally, so growth was not possible. Doing this plate was used to show that the bacteria is not naturally resistant to ampicillin. The second one, with the second to least amount of growth, is LB plus ampicillin plus P-glow. This shows that P-glow makes the bacteria resistant to ampicillin, so growth was found. We did this plate because it was to show P-glow makes the bacteria resistant to ampicillin. The third plate is LB and ampicillin plus arabinose plus P-glow, where arabinose plus P-glow and the bacteria equal glowing bacteria under a UV ray. Growth was found since P-glow makes the bacteria resistant to ampicillin, and we did this plate to show the introduction of arabinose to a plate consisting of P-glow, or has P-glow with the bacteria, makes the bacteria glow. And then so the fourth one with the most amount of growth is LB minus P-glow. Growth was found since there was no ampicillin, and we use this to show that bacterial growth of E. coli is possible just naturally.
Arabinose is a 5-carbon sugar that can be used by E. coli as an alternative carbon source. The enzymes necessary for the metabolism of arabinose are coded by the arabinose operon. There are 2 promoter sites, PC and PBAD. There are 2 operator sites, O1 and O2. The inducer site of the operon is arabinose 1, and arabinose C gene is a gene that contains the arabinose operon, that controls the arabinose operon positively and negatively. It also encodes a regulatory protein. Its binding sites are O1, O2, and arabinose 1. In the absence of arabinose, the regulatory proteins bind to arabinose 1 and O2, causing the operon to fold on itself, blocking the transcription of arabinose B, A, and D. CRP acts by promoting the rearrangement of the arabinose C gene when arabinose is present. Differing from the arabinose operon, the DNA code of P-glow plasmid consists of both the PBAD promoter and the arabinose C gene. Unlike the arabinose operator, arabinose B, A, and D were replaced by a single gene that codes for GFP. When RNA polymerase binding is promoted, more GFP is produced. When arabinose is absent in arabinose C no longer facilitates the binding of RNA polymerase, and GFP is not transcribed.
26% of the world's corn and 86% of the United States grown corn are GMC, or genetically modified crops. GMC corn has new genes introduced from the bacterium Bacillus thuringiensis, which allows the corn to produce Bt protein, a natural insecticide. When this corn was first introduced, many saw it as an alternative to pesticides.
Alright, so we're going to be determining the transformation efficiency, T. T is determined by the number of cells affected by the transformation divided by the amount of DNA. Now the number of cells is easy to figure out. You can simply count the colonies. We got 267 separate colonies on our plate. So we know that it's approximately 267 cells that originated and were affected by the transformation. Amount of DNA is a bit harder to figure out. First we need to determine, fill that in. I forgot about that. First we need to determine the total number of DNA in all of the cells we did. And that was volume times concentration. Volume was 10 microliters. Concentration, I told by packet, is 0.08 microgram per microliter. Which is 0.8 micrograms of DNA. Now the fraction of the arabinose plate that received, the fraction of the DNA the arabinose received is determined by the fraction of the liquid received plate. In this one, 100